Bonjour, je suis Dr. Mikhail Tswett, professeur de chimie ici à Saint-Pétersbourg.
Je suis Dr. Daniel Habanova et j'ai adoré dans cet expériment.
Aujourd'hui, j'ai rencontré de mon invention écrivant des Tswett.
Nous allons montrer comment nous séparons les pigments des feuilles verts.
Nous bouillons 2 kg d'épinards et kérosène pour c'est sûr, et puis faisons évaporer les plus pour déferson.
Maintenant, nous avons cette liquide vert que nous plaçons sur les.
Pour séparer, je verse la liquide dans cette colonne.
Dans la colonne contient une argile blanche, c'est la stationary phase.
Les pigments séparons avec une mixture d'alcool et kérosène. C'est une mobile phase. Okay. 6.
Now that you've finished your extraction of the pigments out of the spinach leaf, you'll be preparing your TLC plate to spot and separate the different pigments of the plant. When you first grab your plate, you'll notice two sides, one shiny and one porous. Don't use the shiny side, people will laugh at you. The porous side is the stationary phase, that's the silica gel. Once you've selected the right side, you want to pick the right edge. The right edge is the machine cut edge. It will be the straightest edge, and there will be no breaks in the silica gel. This will ensure that you have equal absorbance all the way across the edge of the mobile phase that you'll be running through the plate. After selecting the correct edge, you're going to want to mark 1 centimeter up on both sides of the plate for your origin line. Make sure to use a pencil and mark very lightly. Do not use a pen because this will interfere with the absorption of your pigments. You connect these two lines very lightly, and starting from one edge, you mark 6 spots every 1 centimeter. These will be where you'll be spotting your 5 standards and the extract that you've just completed extracting. We are now ready to spot the plate.
Take 6.
We are now ready to spot our TLC plate. You'll notice that we have the extraction solvent from earlier in front of us, as well as the 4 pre-made standards of beta-carotene, chlorophyll A, chlorophyll B, and xanthophyll. It is important when you spot each of these onto your plate that you make a diagram in your notebook so you know which spot belongs to which solution. We'll begin by spotting your extraction solvent from earlier. You'll dip your handmade spotter into the solution. Make sure you don't get it in too deep so that you don't get any magnesium sulfate in the spotter. You'll spot each spot several times with the same solution to make a small concentrated spot of each type. After doing your unknown, you'll go ahead and grab the beta-carotene and spot 4 times on the next spot.
Take 3.
We are now ready to prepare our TLC tank. We'll take 10 milliliters of the magic mobile phase and we'll put it in our TLC tank. Replace the lid on the tank as quickly as possible. We are now ready to develop our TLC plate. After the plate is spotted, it should look like this. You'll notice that the spots are about 1 centimeter above the bottom of the plate. This is so when we put the plate into the TLC tank that the level of the magic solvent is below the level of the spots. You'll notice that the solvent starts moving up the plate via capillary action. You should allow it to run until it's about 2/3 of the way up the plate. Now that the TLC plate is developed about 2/3 of the way, it's time to take the plate out. It's very important when you take the plate out to draw a thin pencil line across the solvent front.
Alright, the last part of the experiment, you're going to be mimicking Tswett's work he did 100 years ago when he first separated the pigments of plant leaves. Doing this, you'll take 30 mils of your magic mobile phase and 3 grams of the silica gel. You pour the 30 mils into the silica gel, and you want to mix the slurry up really good. You have to stir for about 10 seconds or so, so it's nice and mixed. And then you will pour it down your column, making sure to stir while you're doing this so as to get it all in one go. Once you see that your stationary phase is settled and you have the mobile phase sitting on top, it's now time to add about half a mil of the supername of your extract to the top of the column. Once you see that the green color has started entering the stationary phase, it's now time to take 10 mils of your magic mobile phase and pour it down the top of the column. After it's been separating for a while, you can see the beta-carotene, the chlorophylls, and the xanthophyll represented by the yellow, green, and second yellow bands in the column.